Hello, good afternoon, everyone. We're gonna go ahead and get started. Today we will be working through lecture 7, and let me just get the PowerPoint going here. Bear with me, I'm still a little bit sick, so my voice is not really there, but I'll try to get through this. Lecture 7 is about protein synthesis. And in protein synthesis, of course, we know it started at the DNA level where the instruction is contained. And previously, we learned how that information is copied into messenger RNA. And then through the process of translation, we are going to be creating the proteins. So the main objective of today's lesson is for me to teach you how to read the code from DNA. If you're given the ACTG, how do you convert that into amino acids for building the proteins? So let's start with transcription. But before that, there are a few terms that I need you to understand. First of all, when we say genome, genome refers to all the genetic information um, that an organism contains, all the ACTGs in its DNA collectively is known as the genome. As an analogy, you can think of the genome as the entire textbook. Okay, and within that textbook, we have various chapters. Okay, chapter 1, chapter 2, chapter 3. Each of the chapter would represent the chromosome. Okay, so in humans, we have 23 chromosomes. Chromosome 1 is like chapter 1. Chromosome 2 is like chapter 2, so on and so forth. Now, within the chapters of the book, you will have sections in it. Right? Typically, a section is going to tell you information about particular topic. Okay, so 1.1 is going to tell you about chemical fundamentals. 1.2 is going to tell you about functional groups, so on and so forth. So these sections are like the genes on the chromosomes. Each chromosome has multiple genes, and the genes is going to give you information and instructions on how to make the proteins. Okay, so hundreds and hundreds of genes on each chromosome. Now, where do you find these genes on the chromosome? The location of the chromosome is called the locus of the chromosome. Okay, it's kind of like the page number. Right? And the locus, the location of the gene for everybody is the same. Right? Last class we learned about the BRCA1 gene, right? The breast cancer one gene. It's on the same place as it is in my genome as yours. Okay, so by identifying the location of these genes, um, it would enable us to find these genes and to study them, and in some cases, we have to clone them as well. Okay, um, let me try to draw this out for you. Maybe it makes more sense from that perspective. We have chromosome 1 from mom and chromosome 1 from dad. This is not new information, right? Each of our chromosome, half of it we inherit from one parent and the other half from the other parent. So again, this is chromosome 1 and chromosome 1. You know what? Let me just draw a few more for you. Hey, maybe you have chromosome 2 here, which is a little bit shorter than 1. Chromosome 2, and then we have chromosome 3 and chromosome 3 right here. Okay, so 2 and 2 and 3 and 3, and they get progressively smaller as we move towards the higher number chromosomes. So 23 is really small. Okay, and XY, sorry, 22 is really small, and XY is kind of in their own category. But the point is, number 1 from mom and number 1 from dad, they are the same length, same size, and they contain the same kind of genetic information. We call these homologous chromosomes. Okay, so number 1 is homologous with number 1. Number 1 is not homologous to number 2. Okay, 2 is homologous to number 2. A way to think about this is to imagine you have 23 pairs of shoes, right? And you just randomly throw them on the floor. You will still be able to match them up, right? You can find the left shoe and pair up with the right shoe, right? It's because they look similar enough, right? So in the same sense, we can always pair up the chromosome one with the other one, even the chromosomes are kind of all over the place because they are so similar, right? So same length, same size, and same type of genetic information. Now, I want to emphasize same type, not exactly the same. Right? What does it mean? Well, at this location of the gene, maybe you will find, at this location of the chromosome, maybe you will find a gene that encodes for eye color. But let's say your mother has brown eyes, and then your father has blue eyes. Then you might inherit the brown eye gene from one parent and the blue eye gene from the other. They are both genes for eye color, same type of genetic information, but in this case, the version of it, the variety of it is not necessarily the same. They could be the same. Okay, maybe both of your parents have curly hair. So you will find the curly hair locus right here, and this is also the curly hair location, right? And it happens to be the same, right? So the order and the type of genetic information would be identical for the homologous chromosome, but the version of it might not be the same. So different version of a gene is called an allele. Allele. Okay? We use alphabets to represent the alleles. They could be the same over here, same allele, whatever it represent. Could be like I said, eye color, blood type, whether your earlobe is dangling or not, whether you can curl your tongue or not. All these things are genetically determined by the autosomal chromosomes, number 1 to 22. And they could be the same between the homologous chromosome, or they could be different. It all depends on what your parents have, depends on their traits. Obviously, people who are um, same ethnicity, they would share more similar alleles, right? Like both of my parents have brown eyes, okay? So I inherit probably the brown eye allele from both of them, okay? But there will be differences. So that's homologous chromosome, okay? They are same length, same size, same information, same type of information, but not necessarily identical. Previously, we talked about the central dogma of biology. We start with DNA in the nucleus, and we are going to make a copy of the DNA in the form of RNA. But before, it was a simplified version that I told you about. Actually, it's a little bit more complicated than that. Before the DNA can turn into the messenger RNA, the initial copy that you've created from the DNA is actually called pre-mRNA. Pre as in premature, and it's not really ready for use yet. Okay. And then what happens is you will take the pre-messenger RNA, you will process it a little bit further in what's called post-transcriptional modification. It basically helps improve the stability of the messenger RNA, and then it will leave the nucleus through the nuclear pores, and then you will be able to do translation in the cytoplasm. So we're just going to take a closer look at each of these steps today, and hopefully it will make more sense to you afterwards. Just to recap, the reason why we have to do transcription, uh, as opposed to just take the DNA and use it directly to make the protein, is because DNA are too big and they cannot leave the nucleus, right? If they leave the nucleus, they might break down, right? They might suffer damage, and we of course do not want to damage our DNA. And instead, what we want to do is we want to produce a copy of the DNA and in the form of messenger RNA. In addition, this allows us to add a level of control to the to making a protein or not. Listen, our entire genome is like a really thick recipe books to make all these proteins, okay? But we do not need to make all the proteins all the time in every single cell. It's a waste of resources, and we frankly we don't have enough rooms to hold all these proteins anyways. So we only make what we need when we need it, and only in the cells that is supposed to use these proteins, right? And so by having the process of transcription, you are able to add a level of control to it. You can decide when to turn on the gene and when to turn it off. To turn on a gene means you will be converting it into a protein product. And to turn it off means we just kind of leave it on the shelf, not going to use it at the moment. We can use it when we need to. Of course, you know about the base pairing rule. A pairs with T, G pairs with C. Same rule applies to DNA and RNA, where A pairs with U instead of thymine, and G pairs with C. So where does transcription take place? Transcription takes place in the nucleus of the cell. That's where the DNA is. And instead of using DNA polymerase, which builds DNA, we are using RNA polymerase. Okay, RNA polymerase is used to build RNA. The name kind of tells you what it does, right? And it will be using DNA strand as a template to help create the messenger RNA. At the end of the process, you will get a premature messenger RNA. Premature. That's what the pre stands for. And this is not ready for use yet. We have to fix it a little bit. We have to increase its stability before we allow it to leave the nucleus and go into the cytoplasm. Now, this whole thing is, uh, well, this is a section of the DNA, and the DNA is really long, right? This really continues both ends, okay? And the whole thing is called a chromosome. And on the chromosome, in the chapter of the book, you have multiple sections, right? Subsection 2.1, 2.3. Similarly, on each chromosome, you would have subsections, and a section of it is called a gene. Now, just want to clarify what exactly is a gene. A gene contains instruction to make proteins. Some genes can help, some genes help regulate other genes. Okay, so that's what they are. They are instruction to create these proteins, and sometimes these instructions help other genes to turn them on or turn them off, or to modify them. Okay, so that's basically what it is. Everything that occurs in the body is pretty much as a result of the expression of your genes. Okay, it's just a segment of DNA, and this DNA code has the ability to um, produce the proteins for us. Now, if I zoom into these DNA, it would not be nothing more than a bunch of, you don't have to write this down, okay? It would just be a bunch of DNA, ACTGs, okay? So, how do we know when does the gene begin? How does the body know when does the gene begin, right? Like it's just a bunch of ACTGs. Well, it turns out a gene is marked by a special sequence at the beginning. It's still made up of nucleotides, but this special sequence is called the promoter sequence, and it marks the beginning of the gene. Typically, the promoter is really rich in adenine and thymine. Okay, if you recall, adenine and thymine only has only has two hydrogen bonds holding them together. It is weaker than Cg bonds, right? Cg has three hydrogen bonds holding them together. So in this case, um, because we need to gain access to the DNA and we need to open it up, it makes sense to have a lot of A's and T's, right? So it's easier to create the bubble and allow us to gain access to the nucleotides. Okay, so that's the promoter region. So the first step in transcription is called initiation. It involves the RNA polymerase binding to the promoter, unzipping the DNA, and then start the copying process. Okay. If I give you a strand of DNA and I ask you to identify the promoter, you would look for a section that has a lot of A's and T's in it. Okay. And then we have elongation. Now, this part is hard to understand. I will draw it out for you and hopefully it will make more sense when you look at it from a different perspective. Just give me a second here. Okay, so we had the we have the DNA right here. Okay, this is the second step. Our first step was initiation. This step is elongation. Elongation. And what happens is the polymerase will unzip the DNA, and this is an exaggeration to show you what it looks like. Okay, and then there are more DNA ahead, of course. More DNA. You get the idea, right? There we go. Okay, so this is DNA that has been unzipped. Now this is the hard part to understand. Let me just write some nucleotides for you. Of course, they have to be complementary on the other side. Now, just for the sake of completion, I suppose. Okay, I'm going to add a little bit more DNA here in a different color. And that would have been the promoter. Okay, that marks the beginning of a gene. It's like reading a book, right? How do you know you are at the beginning of a new chapter, right? There's like a new title, maybe there's some pictures, right? It says like chapter three. Right? It tells you it's still letters, right? It's still alphabets, nothing special, but it signals you that this is the beginning. So in a similar sense, the promoter sequence have all these A's and T's in it that would tell the body this is the beginning of the gene. And if you're gonna get the recipe to make the protein, you gotta start copying here. Okay, let me know if the next part makes sense to you. Only one of the two DNA strand contain the code for making a protein. This is the coding strand. Okay, coding strand. The coding strand is the one we need to make a copy of, right? Remember DNA are double helix, RNA is single strand, right? Double strand for DNA, single strand for RNA. So I can only be copying one of these strands. I cannot copy both. So which one am I copying? Well, only one of them actually contains the instruction to make the protein, and we call that the coding strand. You cannot tell which one is the coding strand. I mean, I cannot tell either, all right? But in a question, I will tell you which one it is, okay? And it's just hypothetical, okay? Let's say this one is the coding strand. Once again, no one can tell whether it's coding or it's not coding just by looking at the ACTGs. All right, you need other information. But on the test, I will tell you specifically which one is coding. Now, this next part is the confusing part. I am hoping that you are copying the notes. And when you're copying the notes, you are copying exactly as it is written on the screen, right? Like only one of the two DNA, then you copy exactly as it is. Okay, only one of the right? Okay, so for us, copying is it means copying it exactly as it appears. Okay, but copying is a little bit different for polymerase. We already learned about DNA polymerase previously, and so don't copy this next part. Just listen, pay attention. Okay, if the polymerase comes and is trying to copy the coding strand, it cannot be using the coding strand to copy the coding strand. If it uses the coding strand to copy the coding strand, it will look like this. Okay? Let's say the RNA polymerase is here. Don't copy this. Just watch. Okay? This is RNA polymerase. Someone tell me, what letter am I going to put here? The RNA polymerase sees a C over here. What letter is it going to put in that slot?
G.
That's correct, Natalie. It's going to put a G here, and it's going to put a G and a C and so on and so forth, right? Because polymerase can only build using base pairing rule, we cannot use the coding strand to make a copy of the coding strand. If we did, the copy that we get is going to be different from the coding strand. In fact, it's going to be the opposite of what we want. Do you all understand that point? If not, ask me some questions.
Can you tell this again?
Sure. Okay. The polymerase, when they build nucleotides, they can only build using base pairing rule. So when they see a C over here, they are not going to be able to put a C just like that because C and C do not pair up, right? Only C and
Yeah.
G can pair up. So when they see a C, they will put a G. When they see a, they will put a T, or in this case, uracil, and then A, and then C, and then G. So if we use the coding strand to build the messenger RNA, the messenger RNA, which we will need to make the protein with, is actually not the same as the coding strand. It's the opposite of the coding strand.
Yeah, yeah.
Yeah. So what do we
So
do?
if it is copy, then C should produce C, A should A, T should T, G should G.
Exactly, right? So that is the exact reason why we cannot use the coding strand to build a new strand. And instead, we will be using the opposite strand, which is called the template strand. Okay. So let
Yes.
me write out for you. the strand that is used by RNA polymerase to create the premature RNA is called template strand. Template strand. So let's go through this mental exercise together. Okay, we have this. Don't draw this. We have the RNA polymerase coming from the promoter. It's gonna try to copy the coding strand, and then as it moves along, it sees the A here. It's gonna put a U. Remember, no thymine, just uracil. And then we would have A, so on and so forth, following the base pairing rule. So on and so forth, right? So I'm gonna draw the polymerase here. It's gonna keep going, of course. This is RNA polymerase 3. There we go. So by copying off of the template strand, the resulting free mRNA is actually the same as the coding strand. You
Yeah.
see? Right?
Yes.
The information is the same, except what? There's one,
Except U and T.
yeah, very good. Everything is the same, but the T is replaced by uracil because on RNA, there are no thymine, right? We have to use a substitute. We have to use uracil, okay? Another thing to kind of note here is, again, we're talking about the elongation process. As the RNA moves along the DNA, moves along the gene, it will unzip the DNA ahead of itself. And when it's done, it will rezip it at the back. Okay, don't confuse this with DNA replication. There's no rezipping in DNA replication, right? You got to unzip all the way until the end. But here, we are just trying to copy the DNA, and afterwards, we have to put it back in place so that it's safe and not going to be altered in any way. That's elongation. Until we get to the terminator sequence, which is located at the end of the gene, then the RNA polymerase will detach itself from the RNA, from the DNA, and then you will also be releasing the premature messenger RNA. So you start at the promoter, you copy the gene, and then you stop at the terminator. Now, for those of you who might be a little bit confused still, and maybe you don't want to ask questions, that's fine. Here's another analogy to help you understand. When people make a coin at the mint, the mold that they use is not the same as what you want the coin to look like at the end. If it looks exactly like the way you want it to come out, then it actually will come out backwards, right? The mold has to be a mirror image or like an inversion of what you want. And that's how it is with the transcription. We are not going to be copying directly from the coding strand. It will come out backwards. That's why we are copying from the backwards, from the template strand, so that it comes out correctly. Okay? Any questions before we move on?
As it tights up again, so the single strand binding protein is not working.
This is no single strand binding protein, right? Single strand binding protein is only for DNA replication, right?
Okay.
In this case, it's in fact, we don't want the single strand binding protein because as soon as we open it and then we finish looking at it, we need it to wound back up on its own, right?
Okay.
Anything else? Mm. So here comes the questions. On the quiz, you might have to type these things, okay? Whenever you type the answers of these ACTGs, don't type in the space. Whether it's capital or not capital, it doesn't matter, okay? But no space, okay? Just key it in. Follow the instruction in the question, okay? So here it says, I mean, if you don't read the question and you just see this sequence, you don't know whether this is a coding strand or whether this is a template strand, right? But over here, you know what? I'm actually going to copy it to the class notes so I can post it for you afterwards. Nope, that is not what I want. One second. There we go. So here it says, given the template strand, right, like I said, I have to give you that information. Find the coding strand. So that should be easy. If you have the template, then you can find the coding. What's the relationship between template and coding? I'm looking for a single word. Can someone tell me what I'm looking for? It starts with a C. It starts with a C. The two strands of DNA are, you've heard me say it a couple times right now. That's okay.
Copy. Copy, I mean, same.
Complementary is the word I'm looking for. Okay, complementary. The two strands of DNA are complementary, meaning they follow base pairing rules. So if you have A on one strand, the other strand will be T. We're still talking about DNA, right? Both template and coding are on DNA. That's why we're still using thymine, right? Just like that. And what about the mRNA? It technically is pre-mRNA, but you know what? Doesn't really matter. What about the mRNA? What's the first letter? Why don't you tell me? Yeah, we can do a poll. I want to see what people are thinking. Okay, okay. What's the first letter? Don't tell me. I'm gonna launch the poll. Go ahead. What goes here? Okay, anyone else? I'm gonna close the poll in a couple of seconds. All right, thank you. Looks like people are not really understanding it. Okay, which of these strands, the template or the coding, has the instruction to make the proteins? Which one is the recipe, guys? Template or coding? Which one is the recipe?
Template. And
Okay, anyone else?
Coding?
Coding,
coding.
coding, yeah. Coding is the
Coding.
instruction, right? This one is the instruction. The instruction contains the code. The code is the instruction, okay, to make the protein. So when you make the mRNA, you are trying to make a copy of the coding. You're not trying to make a copy of the template, but you are using the template to make the mRNA. Regardless, the end result should be same
It
as the
should be
coding.
T. Yeah, it should
Yeah,
be T.
but
Yeah.
this is RNA, so there's no T.
U.
It would be a U.
It should be just U.
Yeah. Next one is U, and then G. I just copy basically the coding strand. Right? So these two are identical, but T becomes U. Not because of any base pairing rule or anything like that, but because there are no thymine in RNA. So instead of thymine, you have to be using uracil, right? Anything you want me to clarify before we move on? Okay, don't worry, we're gonna have lots of practice, right? And you need to know this before the test. And that's why you probably need to have some papers, blank papers, with you on the test, right? So you can work it out before you type it in. Okay, so let's try again. This is the template, and I'm asking for the mRNA. Okay, you can go from template to mRNA directly if you want. If you need to write the coding first, that's okay as well. I'll give you I'll give you 1 minute to try it, and then we'll take it up. If you have an answer, feel free to type it in the chat box for me. Okay, remember there should be no T in the answer because we are talking about RNA or RNA here. Sorry, guys. I already taught 3 hours this morning, so I'm at the end of my of my line here. Okay, so I have a couple answers coming in. Let's take a look. Look, the template is the one that we are building off of. And when you're building off of a strand, you're going to be using base pairing rules, complementary nucleotides. So if you see an A here, it is going to be U. And then T is going to be A, G, A, U, C, U, nope, C, A, U, and then C, G, U. That's the correct answer. What's the relationship between template and mRNA? Complementary. Complementary. Okay. I will show you what the questions might look like on your test. Give me a second. It's really slow, but it's coming. it would be like this, okay? It would be like in the form of a fill in the blanks, perhaps. Okay, so write the correct mRNA sequence, one codon per blank. Like obviously you're not gonna have the answer filled in for you, right? So like, you would only have the template DNA strand here, and then you would have to key in the correct answer. So if it's ATG, because it's template, it's gonna be UAC, right? So on and so forth. So it says one codon per blank. Don't type in everything all at once into the same blank. Okay? Here, I'll leave this one in the notes for you to use as a reference. Okay, obviously, don't memorize the answer. It's could be a different sequence. And if I give you a coding DNA, then you would do it differently as well, right? Let's take a look at the next one. Okay, this one is the coding. And I'm looking for the mRNA. You should be very happy when you see the coding strand in the question as opposed to the template strand. Because what's the relationship between coding strand and mRNA? Someone tell the class, please, what's the relationship between coding and mRNA?
Copy.
Ooh,
Just copy. Same.
same, yeah. Identical, right? They are the same. That's why you're trying to make the copy. Except the T becomes the U.
You.
So you just copy everything. But anytime you see a T, you change it to U, right? So what's really going on? Here's the cheat sheet. Not that you can use it, but it will help. If I have coding, and there are only four possibilities, right? A, T, C, G. The template will be TAGC, and the mRNA, what's the mRNA here?
Uh, AUCG.
Yeah, that's it. It's just like this, guys. Just like that. Okay, keep this with you. I have one more role for you later on. Not yet. That's gonna take some time. I don't know if we're gonna get to it today. We might. Are we okay? You guys with me?
Yes.
Okay, thank you. So back to the PowerPoint slides for a second. Summary. Coding strands and mRNA are identical. They are the same, except uracil becomes stymied. Template strands and mRNA are complementary, meaning they follow base pairing rule. So ATCG pairs with UAGC. If you have trouble coming up with the RNA from the template strand, you can always convert the template strand into coding strand first and then copy it. Right? It's gonna take more time, but better be safe than than to get it wrong, right? Okay, let's try a poll. Please participate if you can. This will tell me if you actually understood what I explained to you. Like, I mean, this question, whether you get it right or not, will help me understand whether you know it or not. Unfortunately, I only have 6 answers coming in. All right. So this is the template DNA. And for the template DNA, we have to use complementary, right? Base pairing rule to find the RNA, right? So complementary to sorry, I gave you the answer. Complementary to TTA is AAU. The only one that starts with AAU is this one. Okay. GCA, CGA is GCU, and then ACT would be UGA. That's the right answer. Okay. For those of you who didn't answer or you got it wrong, do you understand now? Please ask me if you don't understand. Otherwise, you will lose a tremendous amount of mark on the upcoming quiz and subsequent tests. All right. Different version of a gene is called an allele. We know that already. So just gonna skip through that one. Okay. Now that we have created the pre-mRNA, we are going to do post-transcriptional modifications. Okay, post-transcriptional modifications. Let me write the notes for you here. The purpose of post-transcriptional modification is once again to actually remove some irrelevant information and to increase the stability of the pre-mRNA. Okay, so remove non-coding sections and increase mRNA stability. So let's say this is our pre-mRNA, and I'm going to use a little bit of different color here and there. We have the 5' end and the 3' end. I did not mention 5' and 3' in the earlier notes when we do elongation. It doesn't matter, but here it matters. Okay, so it turns out not everything that we've copied is actually part of the instructions. Right? Only some of it are useful instruction. So in this case, these lighter blue portions, they are called exons. Exons, they are coding regions that will be expressed. Okay, so exons are expressed in the proteins. They are going to be converted into protein. In other words, they're going to be used to make the proteins. And then we have these non-coding regions. They are called introns. Non-coding regions. So the first step in post-transcriptional modification is called RNA splicing, which involves the removal of introns. They don't code for anything anyways, so they're just like fillers. We remove the introns and connects the exons. Okay, so afterwards, we're just going to have one nice one. It's shorter than before, but no more fillers. Okay? Think of a magazine, I guess. Okay, magazine. Like there's a lot of useless stuff in there, right? Like advertisements we don't really need without compromising the content of the magazine. So like you can remove those, right? Uh, those would be like the introns. So now we have the uh, exons only. Okay, this might be exon 1, exon 2, exon 3, and exon 4. So afterwards, they will be all stitched together. No more introns. Now, the next step, number 2, is to add a guanine. Guanine is G, right? Guanine cap on the 5' end. So like there's a special G that they can add over here. You don't have to worry about what's so special about it. But it kind of caps the 5' end to protect it. And then number 3 is add a poly A tail, which is exactly what it sounds like. It's just a bunch of adenines. Okay, approximately 50 to 200. Longer tail equals longer um, longer half-life, or we'll just say it could last longer in cytoplasm. Okay. These RNAs that we're making are not permanent. After they go into the cytoplasm, the ribosome can read them and start to build protein from it, but eventually they're going to break down. When we try to make a protein, we are not making the protein forever. We're just making it for a short period of time. Once we no longer need the protein, we don't want to keep making it. It's a waste of resources, right? So the poly A tail helps you kind of control that a little bit. If you want to make this protein for the next, I don't know, a day or two, sure, we're gonna have a longer poly A tail. But if it's a protein that you only need for like an hour or two, then we'll have a shorter poly A tail. Okay? So 3 things for post-transcriptional modification. Removal of introns, connect the exon together, and then add the G prime cap on the 5' end, and then the poly A tail on the 3' end. And all that is kind of depicted in this picture here. Let me just walk you through it quickly. We have the transcription happening here with the RNA polymerase. The bottom strand here is the template, as you can see. And then the top strand is the coding, just like I drew in the notes. You have the RNA transcript here. And if you take a closer look at the RNA transcript, you can see there are coding exons, and then there are introns. In RNA splicing, we remove the introns, and we stitch together the exons. So only the bright pink. You add a 5' cap and a poly A tail. Then it's going to go into the cytoplasm. And then we're going to be doing the translation afterwards. Okay. I'm just going to give you the answers. You can go through it yourself. Okay. Beginning of a gene, promoter. What do we add on the 3' end? Is the poly A tail. The A is the adenine. And what is removed? We are removing the introns. Okay, so we won't be long. I'm just gonna go through a few more slides with you, and then I think we're gonna stop. Okay. Over here, translation. Just a quick review for you. Nucleotides are made up of monomers of DNA. Nucleotides are monomers of DNA and RNA. We know that already, ACTG. For proteins, the monomers are amino acids. So there are only four nucleotides in DNA, but 20 amino acids in total. Then how do we code for all the amino acids? How can 4 letters, ACTG, codes for 20 different amino acids? So here is how. It turns out every 3 nucleotides forms what's called a codon. And each codon will code for an amino acid. So let's say this is the template DNA. After transcription, I would have the A would become a U, C becomes a G because of the base pairing situation, right? So these three nucleotides makes up a codon, and UGG codes for the amino acid TRP. These are all short forms. They all stand for something, okay? TRP is tryptophan. And then the next one, UUU, it codes for phenylalanine. And then GGC codes for glycine, and so on and so forth. So how many codons are there in total? Well, if you think about it, 3 nucleotides makes up a codon. How many possibility for the first blank here? What letter can go into the first blank for the codon? What are the choices for the first blank? Well, it could be A, C, G, or U, right? So 4 choices. The next blank is also 4 choices, A, C, G, U, and also 4 choices, which means in total it's 4 times 4 times 4. That's 64 different codons. So the 64 codons are all listed on the codon chart. Okay, so this is the codon chart. And the codon chart will be given to you on the test when you're doing it. Okay. Maybe give me a moment. I'll show you how how it would look like in case you are not familiar with it. But I do need a moment to set it up. So please bear with me. Okay, it should work. Let's see. I'm going to switch to student view. This is not your course, so this is just my sandbox for experimenting things. So let's say there's going to be the quiz that you're going to be doing. And then when you start, um, there's going to be the codon chart. And so you can open the codon chart and you can actually download it by pressing this button. Okay. Now on the test, after you click on the download, it's not going to go to your computer because you're using lockdown browser, but it will actually pop up as a new tab. Okay, when you're using lockdown browser. So you will still have the test right here. You'll be doing the questions, and then when you need the reference, you can just click on the tab at the top. During the test, the second tab is going to be the codon chart. Do you understand? Okay, like what you have to click.
So after downloading, it will be the next tab.
It After you press download, it will automatically open a new tab and then it will be over here. I cannot show you right now because I cannot use Lockdown Browser at the moment, right? But on the test, when you click download, it will automatically open up a tab and then the chart will be there. Do you understand?
In chemistry exam, we did this.
In the chemistry, exactly. It's just exactly same as the chemistry exam.
Yeah.
Okay. We actually figured set this thing up together. So it's gonna be okay. All right. So how do you use this chart? I'll show you in a second. But this chart shows you all 64 codons. Okay. Some of them codes for the same amino acid. Okay. Now, even though you have the chart with you on the test, and even though you shouldn't memorize the whole chart, there are 4 that I want you to memorize because it will help you do the question faster. For example, AUG, like August, right? It's kind of sound like August. AUG is the start codon. Whenever we try to start to build a protein, it will begin with AUG because that marks the beginning of the protein. And on the codon chart, AUG codes for this amino acid called MET, methionine. Okay, so all the protein sequence will start with the MET. Does that mean you don't have MET later on? Not necessarily. You can have MET in the middle of the protein, but MET is always going to be the start codon. It's always going to be at the beginning. Think of English sentences, right? The first letter is always capitalized. Does that mean you cannot have capital letters in the middle of a sentence? Of course not, right? You can have a name, you can have a place, and you will capitalize it, right? So MET will show up at the beginning for sure. It might pop up in the middle. As you build the proteins, you will add the amino acids, and then you will come to us. You will know when to stop because it will go. The ribosome will reach one of the stop codons. Okay, you should memorize the stop codons. Like I said, because certain questions it will take too long if you have to look them up. Stop codons are UGA, UAA, and UAG. And you can memorize it like this. This is like a cheesy way I came up with to help students remember. Okay. You got A in biology. You are awesome. And you are good. Right? So these are the stop codons, which marks the end of the translation process. Okay? We are done building the proteins. We don't have to keep going anymore. Let's put this to practice and see how it is done. Okay, so we have the codon chart over here, and we're going to try to figure this out. You know what? I'm going to do this on the notes so that you will see it better. Nope, that's the wrong one. No, it's not working. Hold on a second. Exit. Okay. You know what? Maybe I'll copy the chart over so you see the exact same one. I put this one in the PowerPoint because it's like smaller. It's easier to fit. But I guess I better use the same one for you on the test as the test. Do you guys have this chart? Okay, if you don't have one, you need to print it out for practice when you're studying. Okay. We have we have the first letter here, second letter, and the third letter. So we are trying to decode AUG, which is the start codon. So you should already know start codon is MET. But like, how would you look it up, right? First letter is A. So in the first letter, I look for A. That means it's going to be one of this row. And then we're going to look for U for the second letter. So U is right here. which means it's going to be in this column. And then where's AUU? AUU is right. Oh, oops, sorry. We're looking for AUG. AUG is right here, which is MET. Okay, so one more time. Let's look up AUU. Look up A. We got to find the column first. This is A. U is this column. And we see where they intersect is this box. And in this box, we see AUU. I know it's hard to read this one. It's ILE. ILE. It's just that when you type I and L, it looks the same on the computer, right? IL.
Isoleucine or
Yes, ILE is isoleucine, exactly, right? So you guys don't have to know the full name, but you know, just know that they're abbreviation of a bigger word. Okay. Can you guys please look up CCU for me? CCU.
PRO.
P,
Second,
P, P.
second row.
Yeah. So let's see, right?
Zero.
Second column.
Yeah. So CC. So C is here.
Again, second.
And then second letter is also C.
C.
And then so they intersect here. We're looking for CCU is proline.
Mm, Proline.
Yeah. So keep going, do the rest. If you're stuck, if you don't understand, now is the time to ask. Like I said, if you don't know how to do this, you are not going to be able to do a lot of questions on the test. And when we learn mutation next week, you also need to know how to use the table here. I'll give you a moment to finish the rest. Okay, that's the answer. Is there a particular one that you want me to explain? Are you sure everybody understand it? All right, well, um, I guess that's good news, but it's a little bit hard to believe. So I'm gonna ask you to try this one out. Okay, and you know what? I better copy the question over for you to do. Oh no. There we go. Okay. The format's all wrong, but the question is there. Okay, I'm gonna do the poll. You gotta choose something, okay? That's the only way for me to know if you actually get it. We cannot move on until everybody knows what they're doing. We're doing this question. Anyone else? Okay. Most people got it right. There are some wrong answers, so I'm just gonna go through it one more time. We're looking for CGA. C is right here. G is right here. And they intersect right here. So I'll be looking for CGA in this box, which is ARG, arginine. All right. I'm gonna skip the next concept check question for now. We can come back to it. What else do we have here? We have another practice. You can do that. I want to do this. I want to do example 2 with you. Okay, let me go here, do one more, and then we'll call it a day. I think my voice is tapping out. Last one. So we have template DNA here. Of course, to make the protein, you need to go from DNA to RNA and to amino acid. So we have the template DNA here. Template. And for transcription, we're going to make it into RNA. We will assume this is all exons. Okay, we're not cutting anything out. So what would the mRNA be? And once you have the mRNA, you need to find the protein. The protein. And this is why you have to be very careful and understand what you're doing. Because if your mRNA is wrong, your protein will also be wrong. Okay? So type me the first 3 letters when you have it, please. You can private message me, you can message the whole class, doesn't matter. Probably private message would be good, so other people can do it too. I'm looking for the first 3 letters. I'm not looking for the amino acid. I'm just looking for the first 3 letters. Anyone? Yeah. Okay, so this is AUG, right? Remember, template and mRNA are complementary. So you gotta do base pairing rule. Okay. So AUG, I'm just gonna put a dash here to separate it so it looks a little bit clearer. And then the next one, the next one is CAU, GGA, UUA, UGU. Are we okay? Any questions about any of these? Okay, good. Once you have this, then you can write a protein by using the chart, right? So AUG is MET, CAU, U is HIS. Okay, it's okay if you haven't finished yet. Okay, I'm just writing down the answer for you. So when you practice on your own, you will have the answers. For those of you who have finished it, please help me double check my work. I might be making a mistake that I don't know about. And that's it. Oh my goodness. Sorry, I pulled the wrong thing. Can I get some feedbacks? Are you guys with me? Usually this is the part I would walk around the classroom and then, checking people's work.
Yes,
But
sir. Yeah.
yeah, okay. The rest of you, okay. Um, I'm just gonna quickly do this one for you so you have the answers available to check afterwards. So this time I gave you the coding. When you see the coding, you should rejoice. You should be happy because it's so easy, right? Coding, I don't have to think. I just copy it. But change the T to U. And we have the proteins. That's it. Now you look at this, you might freak out a little bit. Hey, did I do it wrong? How come it doesn't stop? Remember, proteins can be really long, right? And sometimes there are hundreds of amino acids. So this might not be the end. It might keep on going for a while, right? Just because you don't end up with a stop codon, it does not necessarily mean you did it wrong. All right. Okay, that's it, guys. Work on these stuff. The quiz is going to be next Thursday, I believe. Next Wednesday, actually, next Wednesday. We will finish everything of lecture 7 on Monday. If you have questions about these things, feel free to email me. Okay. Thanks for sticking around and bearing with me. Enjoy the rest of your week, guys. Bye bye.